Hi, everyone. My name is Gregg Beckham. I'm a chemical engineer and one of the co-PIs and the institutional lead from NREL in the Agile BioFoundry. It's nice to be here with all of you today on Zoom. So we can go to the next slide, please. So what I'm going to talk about today is the focus of the ABF 4 primary core projects that focus on the production of 2 fuels products and 2 chemicals products. And these are slated to be or intended to be in direct sort of response. And whoops, oh, I don't know, there was a timer on that or something, but yeah, thanks, Angie. um, Towards the US DOE industrial decarbonization roadmap, for which you can see on the left-hand side of the slide that, chemicals and, of course, fuels, as many of us know, are major contributors to greenhouse gas emissions. And so the Bioenergy Technologies Office has goals, which you can see on the right-hand side of the slide, towards decarbonizing transportation, especially through sustainable aviation fuels. And we have GHG emissions and volume targets as noted there, as well as towards decarbonization of industrial chemicals. And you can see they're related to commercial production of 10 plus renewable chemicals with 70% GHG emissions reduction and 1 million metric tons per year of CO2 emissions reductions. If you go to the next slide, please. So one of our first, or I should say, one of our 2 fuels efforts, again, sort of in this technology readiness level that we sit in the Agile BioFoundry, focuses on the production of ethanol through a novel process that goes beyond sort of the standard pretreatment, enzymatic hydrolysis, and fermentation. And so why ethanol? If you see on the left-hand side of the slide, anaerobic processes for fuel production relative to aerobic processes are considerably cheaper, both in terms of capital. You can see there, 60 versus 10 sort of $1 million, and then for installed cost, and then also on operating cost, it's about a factor of 4 to 5 cheaper in terms of operating, especially around agitation and cooling for aerobic versus anaerobic bioreactors. On the top right of the slide, you can see, as of course most folks know, ethanol is quite straightforward to separate from water. That's of course technology that's been around for many years, and ethanol is achievable at high titers and high yields as well from sugars. And then on the bottom right of the slide, ethanol is also very straightforward, and it's very well known how to upgrade that into isoalkanes, cycloalkanes, and other types of compounds that can go into SAF, gasoline, and diesel. The next slide, please. And so the conventional lignocellulosic fuel production process that goes through ethanol is shown on the top of this slide. This, of course, is something that the Bioenergy Technologies Office has supported efforts in and now scale-up efforts in for quite a number of years, where one goes through a pretreatment that can take on many types of different chemistries, and then enzymes are produced that can depolymerize polysaccharides. Those are then added to fermenters, and then eventually some ethanogen is added to a fermenter, and ethanol is produced and goes to downstream processing. What we're focusing on at a sort of a high-risk, high-reward approach in the ABF is this concept of consolidated bioprocessing, where anaerobic thermophilic cellulolytic organisms are used to essentially not use pretreatment, but rather digest polysaccharides out of a whole biomass or delignified biomass through some lignin valorization strategy. And then we go to DSP directly. And so this could eliminate things like pretreatment and on-site enzyme production, which are considerable costs. Next slide, please. And so just to sort of hammer that home, these are potential impacts from the consolidated bioprocessing idea, wherein, looking at sort of what's shown in blue boxes on the left-hand side of the slide, pretreatment, enzymatic hydrolysis, and fermentation and cellulase production are considerable contributors to the cellulosic ethanol production cost, as well as wastewater treatment and the boiler turbo generator for burning lignin. And so the removal of these could be potentially quite advantageous for both economics and not shown here, but also for GHG emissions. And it could also allow for fewer pH swings, which would reduce the burden on wastewater treatment, which is a major uncertainty in process modeling. Next slide. And so what we're working on in the ABF is first screening across multiple cellulose degrading or cellulose depolymerizing thermophiles with focus on called Acellus species. These are gram-positive strict anaerobes. They're some are highly cellulitic. They're among the fastest growing organisms on crystalline cellulose, and they can grow around 70 to 80 C. They have different challenges than organisms that have been pursued for many years in the CBP context, including Clostridia. Um, and right now, what we're doing is trying to identify the best strain, develop genetic modification tools, and engineer its metabolism to, instead of becoming an acetate producer, being an ethanol producer. Next slide, please. We also have a second project in terms of production of alkanes via fatty acids, and I realize I need to speed up a little bit. In this case, we're using Rhodosporidium toluloides. It's a really fast-growing oleaginous yeast. It's also carotinogenic. I can never say that word. But it has a lot of metabolic versatility. And you can see on the right-hand side of the slide that Dee Lu and John Gladden and their team at Sandia have done a lot of work in this space to be able to produce a considerable number of compounds already, including, well, you can see these here, and many at high titers. And so it's extremely engineerable, including from fatty acids. They've been able to produce fatty alcohols at 10 grams per liter, which is quite impressive. Next slide, please. And in terms of the lipid conversion to sustainable aviation fuel work, instead of focusing on cellulosic sugars in this effort, we have been looking at oilseed meal. This is based upon industry feedback. There are these streams in first generation ethanol plants, for example, that could be used to convert into SAF-based lipids. And so Dee and John and their team are working on pretreatment technologies to be able to produce sugars and amino acids, which then can be fed to our toluloides strains that are engineered to produce free fatty acids or lipids that can be secreted, for example, or extracted in situ. And so there's a lot of work going on that's really grounded in TEA and LCA around this approach. Next slide, please. And so just showing you some of the techno-economic analysis work. We've been looking at various scenarios where you would undergo some deconstruction, aerobic bioconversion, and then really sort of what the handle we have in the ABF, the handles, I should say, we have in the ABF are things like the ability to do lipid secretion, or if we can have cells undergo autolysis or have cells secrete lipids, and you can see the estimated cost savings there in terms of the TEA, the process modeling efforts. Next slide, please. And there's a substantial amount of metabolic engineering going on in here. We have multiple, as you can see, many designs for knockouts, overexpressions, and then specific targets for free fatty acids, which would be an ideally secreted product from Rhodosporidium toluloides. Next slide, please. We also have efforts going on in ABF around the production of biochemicals. These, like noted before, and including for SAF as well, are motivated by the potential for offset in greenhouse gas emissions. And if you look at the left-hand side of the slide, bottom left-hand corner, what's shown here is GHG emissions for the production of nitriles and organonitrogen compounds generally. And you can see one of the biggest ones is HMDA, that's hexamethylene diamine. And on the right-hand bar graph, you can see adipic acid has the highest GHG emissions among most mono and dicarboxylic acids produced at 1 million metric tons per year or greater. To this end, we're working on the production of muconic acid, shown on the right-hand side, sort of in this circle diagram. This is a building block that can be converted into hexamethylene diamine, or HMDA. It can be converted into adipic acid, acrylic acid, caprolactam, and it can also be for direct replacement chemicals, and it can also be used as a performance advantaged product in plasticizers, polyacrylates, polyesters, and cross-linked polyesters, among others. Next slide, please. And so we have been working on this since the inception of the ABF and have made considerable progress in the engineering of Pseudomonas putida, KT2440, which is the bacterium of choice that we work on in this particular biochemicals project. We've engineered it to convert glucose, xylose, and not shown here, but also arabinose into muconic acid. And you can see we're at about 40 to 50 grams a liter, about 0.5 gram per liter per hour, on the way to 1 gram per liter per hour. And our molar yield is between 40 and 50% based upon most of our bioprocess development efforts. Next slide, please. We've also done techno-economic analysis and life cycle assessment for the strains and sort of the performance of our strain as we've gone through several years of metabolic engineering and bioprocess development. On the top left of the slide, or left-hand side of the slide, you can see minimum selling price of adipic acid or muconic acid. And you can see that we've been able to drive that cost down now almost close to cost parity, around 270 per kilogram. And the current adipic acid price, as noted in the bottom of the slide, is 250 a kilogram. And depending on what kind of sugar production process you use, whether it's deacetylation mechanical refining, deacetylation dilute acid, or dilute acid pretreatment, you can drive that cost down further, depending on how cheap you can make your sugars. Next slide, please. And we've also scaled this up to 150-liter bioreactors. And you can see that sort of the scalability, at least this is, a couple of years ago now, or a year ago now or so, but at the sort of small scale and the large scale, we're able to get similar titers, yields, and productivities. And we have plans to further scale this up with better strains soon. And then lastly, another biochemical that the other primary platform chemical that we're focusing on is 3-hydroxypropionic acid. This has been worked on for many years as muconic acid in the Agile BioFoundry. And we've been able to reach high titers, rates, and yields in a fungus, the Aspergillus fungus. 3-hydroxypropionic acid is of interest because it can also, like muconic acid, be made into a lot of different compounds that are sold at very large scale, including acrylic acid, methyl acrylate, acrylamide, acrylonitrile, and others as well, as you can see in the middle of the slide. Next slide, please. In terms of efforts in the ABF, you can see on the bottom left of the slide, we established a functional pathway in 2018, and then Kyle Pomeranian and John Magnuson's teams at PNNL, our team at PNNL, have been working to improve the titer, and this is on a log scale, of 3-HP since 2018. And you can see now we're close to 40 to 50 grams per liter, which is really exciting from cellulosic sugars. And you can see again the progression of the techno-economic analysis as we've moved both yield and productivity improvements in Aspergillus. You can see that walk along on the white dots on the MSP diagram on the bottom right of the slide. So next slide, please. We also have done process modeling, TEA and LCA, for acrylic acid production from the 3-HP. And you can see it's quite cool. On the left-hand side of the slide, you're almost to net zero carbon emissions for producing acrylic acid. So 0.13 kilograms of CO2 per kilogram of acrylic acid, which is super exciting. And then the techno-economic analysis is shown on the right-hand side of the slide, and it's currently estimated to be up around $3 a kilogram, a little bit higher than the current, or sorry, I should say the fossil baseline is shown there, but the current acrylic acid price, like adipic acid, has also gone up considerably and is now we're at more or less cost parity. Next slide, please. And going further, we are also, of course, using all of the great capabilities, the ABF, to make metabolic engineering predictions to be able to further improve the titers, rates, and yields for 3-hydroxypropionic acid. And some of our biggest titer rate and yield improvements have come from non-intuitive targets based upon some of the tools that you've either heard about or will hear about presented by others in the ABF.